Hi everybody, welcome back to another video. So in today's video we're going to talk about different blood types and how does blood typing actually work. Okay, so if you enjoy the video, if you learned something from it, please consider leaving a like, commenting, and subscribing. Let's get right to the video. So blood typing, how does it actually work and how is it even determined? Well, a blood type, okay, is determined, and pay close attention here, it's determined by the surface antigen on red blood cells. Okay, but before we actually talk about the different kind of systems that classify these surface antigens on the surface of the red blood cell, we have to talk about what the hell an antigen is. So back when I did my plasma membrane video, you will recall, and if you don't, go and rewatch it, or just skip that part. You'll recall that I mentioned that sometimes there are these things called glycoproteins on the surface of the membrane, and they're part of this kind of giant thing called the glycocalyx, which gives the cell a kind of identity. Okay, well, that's kind of what we're going to be talking about here. Those glycoproteins, or that glycocalyx over there, okay, they carry these glycoproteins on the surface of plasma membranes. Now, in this, video, we're going to talk about the surface antigens or proteins on the surface of this red blood cell. Okay. Now, let's say here's the plasma membrane of this red blood cell, right? It contains this antigen, okay, and it contains, okay, let's say it contains this guy, okay, and it also contains this guy. Boom. Okay, so these right here, these are all surface antigens. Okay, but what is an antigen? Okay, what the hell is an antigen? An antigen, okay, is a large complex molecule, usually a protein, or it could be a carbohydrate, that triggers a specific immune response against itself when it gains entry to the body. Okay, so all of our cells in our body, doesn't matter what it is, they have antigens on their surface. However, our immune system has been trained to not elicit an immune response against those antigens, right? Otherwise, we would have an autoimmune disease, okay? However, when a foreign antigen gets into our body, the immune system will recognize that foreign antigen. It'll say, hey, you know what? This is not from my own body. This is foreign. I need to get rid of it as soon as I can, okay, and as fast as I can. Now, why does this matter when we're talking about blood types and all that stuff? It matters because there's these things that we like to do as humans called blood transfusions. And whenever somebody's low on blood, we like to match their blood type, okay, and then transfuse a blood to them. Now, why does this matter? And how is it related to this antigen stuff? Evolutionarily, our immune system didn't develop knowing that we would have to give blood transfusions, right? It just developed to protect us from any foreign substance. To our immune system, a blood transfusion, aka taking somebody else's blood and introducing it into your own blood vessels, into your system, right, is a foreign entity. All right, that is not known to us. We don't know that thing. And your immune system is threatened by it, okay? It'll immediately try to attack it. Now, these surface antigens, this is what the immune system is eliciting a response against. So, if we could match these surface antigens of the guy who is giving us blood with our surface antigens, then and only then will the immune system not elicit a response against it and not attack it, okay? Because remember, the whole point of a blood transfusion is to give somebody blood who is low on blood, right? More specifically, give them red blood cells, because that's what's carrying oxygen. But if I do that, and my immune system starts attacking those red blood cells, then it'll start killing them. And then we're back to square one with no new red blood cells. Okay? So I have to match these surface antigens of the donor to my own surface antigens, so the immune system doesn't attack it. Now, this surface antigens, and by the way, antigens, they are named like this. They are named because of this: antibody generating. Okay? So the way that the immune system actually starts targeting these guys is by generating an antibody against them. And, in the next video, we'll learn all about how the immune system can make antibodies and all that stuff. Okay? But antigen, antibody generating. Okay? That's where that comes from. But again, antigens are just these large, complex molecules that trigger a specific immune response against the self. Okay? And remember, antigens are foreign to the body, okay? And they can be anything. They could be, for example, proteins on the surface of a bacteria. That's an antigen. They could be foreign red blood cells. Even organ transplants. That's why sometimes they don't last long, or whatever, because they have to match exactly the antigens on the surface of our own organs, okay? All right, so that has to be gone out of the way. That's really important. Now, there are different systems that classify what kind of proteins or antigens we have on the surface of red blood cells. Okay, so many systems. Now, this video, we're only going to talk about the most common and well-knowns, such as the ABO system, and this other one, which is the Fisher race system, or sometimes called the rhesus. Okay, rhesus. Now, you might be wondering, hey, what the hell is this? I've never heard of this. Well, sure you have. You ever seen A positive blood? This positive comes from the Fisher race system. If you've seen A negative blood, the negative also comes from the Fisher race system. Okay? Now, the Fisher race system describes multiple antigens. In fact, the Fisher race system describes upwards of 50 blood antigens. However, there are 5 primary or important ones. Okay, so it starts out with the big D antigen, then the big C, then the little c, then the big E, and then the little e. Okay, so these are the 5 primary antigens of the Fisher race. But remember, this system describes upwards of 50. That means that there's 50 antigens on the surface of this red blood cell, okay, including these guys, and including A, B, and O, okay? Now, I just want to, for the purposes of understanding, clarify, these are not the only systems, okay? There are multiple other systems. There is the Kell system, for example. There is the Duffy system. There is the KID system. There are so many systems that describe upwards of like 100 antigens on the surface of our red blood cells, and that should make sense because this is just too simplistic of a view, okay? However, for the sake of learning, we're going to look at these ones, okay? Now, there's also another reason why we usually learn more about the ABO and the Fisher race system than any of the other systems, and that has to do with their antigenicity, okay? So antigenicity, or their immunogenicity. Immunogenicity. What does this mean? This means that these antigens, these surface antigens specifically, they are the ones that can initiate a very strong immune response, okay? So for example, any of those other systems that I just named, the ones that probably no one knows about, unless you're a hematologist, those ones, they don't initiate as strong as an immune response as these ones do. Okay, that's why these ones are more well known. All right. Okay, so that's that kind of reasoning there. Now, let's kind of give an example. Let's say this red blood cell here, okay, has this surface antigen on it, which is A, and it has this surface antigen, which is B, and it has this one that is big D. What would the blood type of this guy be? Well, he has A and he has B, so we know that he's definitely an AB. What is this D? Is it just going to be ABD? No. The D represents whether or not you are going to be a plus or a minus. In fact, they say that this D, back in the day, it used to be called the RH factor, okay? RH stands for rhesus, okay? So the rhesus factor, or the RH factor, if you have this big D antigen on the surface of your red blood cell, they say that you are RH positive. If you do not have this guy on the surface of your red blood cell, this antigen, then you are RH negative. Okay? So this individual at this current moment is AB positive. Okay, great. Now, and again, in this video, we're only going to be talking about ABO and the D antigen. We won't be covering the C, little c, E, and little e, okay? Great. So let's just kind of get that out of your head. But one note I will say is that an individual who has capital C on their red blood cell cannot have the little c. It just can't happen. It has to be one or the other. Okay. Now, let's talk about something else, which is extremely related to what I was mentioning before. Here's all of the blood types that we're going to be talking about here. We have blood type A, blood type B, AB, O, and whether or not you have the rhesus factor. Okay? But guess what? Individuals that have a specific blood type, right? Their immune system learns that, okay, so my red blood cell has, for example, the A antigen on it. But there are other blood types that are not on the surface of my red blood cell. So, in order to protect myself, the immune system starts making antibodies against those guys. And if they ever show up in our system, then the immune system will start killing them. So what do I mean by this? Somebody who has a blood type A, their immune system starts to produce anti-B antibodies. Okay, so these guys, they have antibodies for the B antigen, all right? Because to them, to their immune system, B is a foreign antigen, right? And if it enters their body, it's a sign of like danger, it's foreign, okay? And so the immune system is kind of making this, okay? And again, this is extremely related to what I was saying before, where I said that evolutionarily, we just didn't develop to, transfuse blood, right? So this is a protective mechanism. What about B? Take a guess. Which antigen are they missing on the surface of their red blood cell? Well, they're missing A. So their immune system makes anti-A antibodies. But this is a peculiar case. What if you have both A and B on the surface of your red blood cell, just like this gentleman? Well, in that case, you're not missing any antigen, and your body doesn't make any. None. Your immune system makes no antigens. And by the way, this is why this guy can be a universal receiver, and we'll see that in the next slide. Now, the other thing is this O blood type. What does O stand for? O stands for 0. That means you have 0 antigens. No antigens on your surface of your red blood cells. Now, again, take this with a grain of salt. When I say no antigens, I mean no antigens of the ABO group, as in you don't have A and you don't have B. But obviously you have antigens on the surface from those other systems, okay? But remember, these two are the most important because they are the more immunogenic. They initiate the strongest immune response. Now, O, what do you think they would have? They don't have A and they don't have B, which means they would have both A So both anti-A and anti-B antibodies. What about D? What if somebody doesn't have this D? Okay, let's say they're Rh negative. Okay, let's say that they are Rh negative, which I said means absence of this guy. Well, so far we've seen that an absence of an antigen results in the production of an antibody. But funnily enough, the absence of this capital D antigen does not result in the production of an antibody. That means there are no naturally occurring capital D antigens. Okay? Now, does that mean that it can't form, not antigens, antibodies? Does that mean that it can't form? Absolutely not. It can. But the only way that it can form, okay, is if somebody who is Rh positive donates their blood to somebody who is Rh negative. Only if this occurs, then an antibody against this D antigen will form. Why is that? Because if somebody who has the D antigen on their red blood cells, right, they transfuse their blood into my system, which does not have the D antigen, my immune system will immediately recognize that, hey, this thing is not ours, it is foreign, okay? And if they recognize that this is foreign, they will immediately start to create antibodies as a form of defense to try and kill it, okay? In that scenario, yes, we will have D antibodies. It's also important to remember that while big C has little c and big E has little e, there is no little d antigen, okay? It just doesn't exist. However, the little d antigen has been used to kind of represent the absence of the capital D. So sometimes you might see that. Okay, now that we got all of this stuff out of the way, let's go through a scenario of a blood transfusion and show you how important it is to match a blood type, okay? So let's say that there is a recipient that requires blood, okay, and their blood type is B. But then the doctors managed to find somebody who they thought had a blood type of B, but they actually have blood type A. So what would this look like? Well, on their red blood cells, they have the A antigen, and they have what antibodies? They have B antibodies. Okay. The recipient, the guy who's receiving the blood, his red blood cells on the surface have B antigens. Okay. And in their bloodstream, this guy has A antibodies. Let's see what happens when this recipient receives type A blood. And remember, this blood transfusion is being done because they need more red blood cells because they are in danger of low oxygen, okay? Because red blood cells carry oxygen. When the recipient receives the type A blood, look what's going to happen. Those antibodies that they had inside of their system that was specific for what? Blood type A, right? Because remember, this guy's blood type B, and blood type B has anti-A antibodies. Those dudes will immediately start to attack the donor's red blood cells. And they will start to kind of clump up to one another and aggregate. That is called agglutination. Okay? Afterwards, a little bit after this, these red blood cells will start to rupture. And when they start to rupture, obviously they start releasing their contents out. And we know red blood cells are super high in heme, right? Hemoglobin, which can actually cause damage to the kidneys as function. Not only that, these guys are now ruptured and they're clumped up to one another, so they can block your blood vessel. And imagine this, you're already low on blood, so that's why you're getting the transplant. But now there's this clump of ruptured cells blocking your blood vessel, which means no blood can go through, no red blood cells of your own can go through and deliver oxygen to the tissues, which means there's a possibility of you dying here. Okay? So this is why it's so important that you want to match blood types between the donor and the recipient, because if you don't, something like this could occur. Now, some of you might be asking me, hey Keon, what about the donors' antibodies? Don't these guys start to attack this? And you're asking a great question, but usually when we do a blood transfusion, what we do is we take the blood out, right? We centrifuge it, so we separate the blood into its different compartments, right? And you must remember from my previous video that antibodies, gamma globulins, or immunoglobulins, same thing, they reside in the plasma compartment. So what we do is we almost entirely isolate only the red blood cell component, and then we transfer it into the recipient, okay? That way, none of these guys actually get in to this dude's system, okay? Alrighty. So this is to kind of make you understand that it's so important to match blood types, and if you don't, bad things can happen, such as this agglutination and this rupturing. Now, in this next slide, what we're going to do is we're going to try and figure out how can we actually figure out someone's blood. So let's say I have a patient come into a clinic here, okay? And they don't know their blood type, okay? And so they're going to give you a sample of blood, of their own blood, right? And we have to blood type it. So how do we do that? Well, what we're going to do is we're going to take that sample of blood and we're going to separate it into three, this is the sample of blood, we're going to separate it into 3 test tubes, okay? 3 test tubes, great. And in each test tube, we're going to test for 1 antigen, okay? Remember, we're assuming that we're only doing the ABO and the rhesus factor. So we're going to test in this one for antigen A, in this one for antigen B, and in this one for antigen D, okay? Remember, there's no O antigen. O just means that there's nothing. How am I going to do that? How am I going to test for the antigen? Well, remember how I said that over here, this guy has A antigen, right? And if we transfer this red blood cell that has A antigen into the blood vessel of somebody who has blood type B, which, by the way, they express antibody A, okay, then those A antibodies will start to attach onto the A antigen and cause this kind of agglutination clumping response, okay? We can actually use this to our advantage when we're trying to blood type somebody, figure out somebody's blood type. I can go out into the laboratory, right, or some sort of pharmaceutical place, and I can buy antibodies that are specific for the B antigen, the A antigen, and the D antigen. Now remember, there's no naturally occurring ones, but I can actually go and buy this, okay? And using that, I can actually blood type somebody, okay? So one more time. We got a blood sample from a patient. We separate it into 3 distinct vials, and in each vial we are going to test for a specific antigen, okay? So over here I'm going to test for A antigen. How am I going to do that? I'm going to add the antibody that is specific to this A antigen. And if I notice that in this test tube, this clumping kind of response of the previous slide occurs, then I can confirm that, okay, the A antibody has bound to the A antigen. And that must mean that this guy is blood type A, okay? Same thing I will do with B, and same thing I will do with D antigen. So just to clarify for you guys, these letters here represent the antigens, okay? So before we get into this example, let's go ahead here and create a little legend for ourselves, okay, so that we can easily refer back to. We can say that blood type A can give to 2 blood types, it can give to A, obviously, and it can give to A and B. Okay? Well, why can it give to A and B? Because remember, let's go to the previous slide. A and B have no antibodies in their system. So if I transfer blood type A into these guys, there will be no adverse reaction because they don't have any antibodies for this agglutination and rupturing response to occur. Okay? So A can easily give to A and B. What about blood type B? Who can they give to? They can give to blood type B and AB. Same exact response. Okay? Who can blood type AB give to? They can only give to AB. Okay? Only give to AB. And blood type O, you guys know as the universal donor, okay? Which can give to blood type A, blood type B, blood type A and B, okay? And blood type O. And the reason they can do this is because they have no antigens on their surface. And if you have no antigens, such as this guy, right? The immune system will have no adverse reaction because it doesn't sense anything foreign. It just sees a red blood cell with nothing on it. So it can't do anything. And remember, the whole point of an antigen is to initiate an immune response. And if there's no antigens, then there's no immune response initiated. Now, this concept of a universal donor, again, is a little bit, misleading because, remember, I said there are so many different blood systems, right? And we can't just assume that, oh, this guy is a universal blood donor, so blah, blah, blah. We have to match most blood systems. But for the purposes of this video, this is enough. Now, this is just the blood groups. What about the rhesus factor? Well, if you are Rh negative, okay? Rh negative, you can give to somebody who is negative and positive, okay? Why can you give to somebody who's negative? Well, because they're the exact same as you, so you can do that. But why can you give to somebody who's positive? Because somebody who's negative, right? They don't have the D antigen. They don't have this guy, okay? Which means that if I give it to somebody who does have it, well, there's going to be no adverse immune reaction because they don't have the D antigen. So if I give them blood that does not have the D antigen, then we're good. The only time you shouldn't do that, you shouldn't transfer the D, is because if somebody has this Rh positive, meaning they have the D antigen, they can only give it to somebody who's positive. They can't give to a negative. No go. And we talked about why. It's because if somebody who carries this D antigen gives it to somebody whose red blood cell does not have the D antigen, then this will cause an immune response to occur and, the development of the antibodies. Okay, so we don't want that. Okay, so this is our map, and we can come back to this. Now, let's quickly go through what I mean here. So, sample of blood, separated into 3 vials, where I'm going to test for an antigen with their specific antibodies. So, let's do this. I'm going to add an antibody that is specific to D. It's anti-D antibody. Okay? And I'm going to add it into this vial. Let's see if the agglutination response occurs. Nope, nothing happens. I'm going to add another antibody. This one is specific, anti-B, into this vial to test for the B antigen. And let's see if anything happens. Okay, nothing happened. Now, I'm going to add another antibody, but this one is called anti-A. And over here, as soon as I add this in, after a few minutes, something cool happens. Watch. Boom. I will get this agglutination clumping response. Okay? Now, this is visible to the naked eye, but you can view it underneath the microscope. Okay? But you can clearly see sometimes that there is like this clumping occurring. Either way, now that we know this occurred and no reaction occurred in these ones, we can conclude that the blood type here is actually A, okay? And since they didn't have the D antigen, it's A negative. Okay. Now, who can this individual receive blood from? Well, obviously it's going to be A negative because same can always give the same. But let's take a look at our chart real quick. Okay, who can give to A? So A can give to A, and O can give to A as well. Okay, so O, but is it O positive, O negative? Well, let's see. This guy has a negative, so only a negative can give to a negative. So O negative. All right, who can this guy donate to? Well, again, he can donate to A negative. But who else? Can he donate to A positive? Absolutely. Absolutely he can. Remember, negative can give to both negative and positive. Okay? So A negative and positive. Next. A and B. Because remember, A can give to both A and B. Okay? And since A is negative here, it can give to both A and B positive and negative. Easy. Easy. All right, so just to go ahead and speed up the process, I've gone ahead and pre-drawn some of the samples, okay? So now let's start with this one, okay? Let's say we got a new sample of blood, and we separated it into those 3 vials, and we want to, again, test to see what kind of blood type this guy is. So we got A, B, and D. Okay, so I go out to the store, or, you know, the pharmacy or whatever, okay? And I buy antibodies for D, and I put them in there, nothing happens. I buy antibodies for A, I put them in there, nothing happens. I buy antibodies for B, and lo and behold, boom, clumping occurs. So, what blood type is this guy? This guy definitely is B, and nothing happened in the D, so he has to be B negative. Okay? And who can B negative give to? Well, B negative can definitely give to B negative. Okay? But who else can he give to? Who can he receive from? Okay, he can receive blood from B negative because he's B negative, and he can receive blood from the universal donor, which is O negative. Okay, alrighty. Who can he donate to? There we go. He can donate to B negative again. He can donate to B positive for the same logic as last time. He can donate to A and B, positive and negative, again, same logic as last time, because remember, if you're B or A, you can give to A and B, okay? And if you're negative, you can give to everybody, negative or positive, okay? Anybody else he can give to? No, this is it. Okay, next one. Again, a new sample of blood I'm given, A, B, D antigen. I go to the store, I buy an antibody for D, nothing happens after I put it in. I buy an antibody for B, and I buy an antibody for A. Okay, and after I put them in, I notice that clumping occurs, or agglutination occurs, in both A vial and the B vial. Okay, so what can I conclude here, guys? I can conclude that this guy is both blood type A, B, okay, because something happened here and something happened here, but since nothing happened here, he's AB negative. Okay, who can he receive blood from? Obviously AB negative. And he can receive blood from A negative, B negative, and O negative. Okay, because remember, A and B can give to both AB. Okay, and negative can just give to everybody. All right, and O negative is a universal donor. Now, who can he donate to? He can give only to AB, but since he's negative, he can give to both AB negative and positive. Easy. Now, again, new sample of blood, A, B, and D. I go to the store, I buy antibodies for D, I buy antibodies for B, and I buy antibodies for A. I put them all in their respective vial. Nothing happens. You know what this guy is? If nothing happened, A and B aren't there, so that means this guy is O. D isn't there, so he's O negative. This guy is the universal donor. He can give to everybody, but he can only receive from one guy. That's O negative. Okay? You can donate to A plus and minus, B plus and minus, AB plus and minus, and O plus and minus. Okay. All right, and that's because literally he has no antigens. He doesn't have A, B, or D, so when you transfuse the blood, no immune reaction occurs. Okay. Alrighty, let's say new vial, okay, A, B, and D. I go out, buy antibodies for D, for B, and for A. What happens? Immune reaction only occurs in D and A, which means this guy is only A positive. Who can this guy receive blood from? Who can he receive blood from? He can receive blood from A positive, of course. He can receive blood from O negative, okay? He can receive blood from A minus, because remember, minus can give to everybody, okay? And O positive. Why? Because O can give to everybody, and positive can give to positive. Easy. Who can he donate to? Who can he donate to? He can only donate to A positive and AB positive, okay? Because positive can't donate to negative. Easy, easy. Okay. Now, I would suggest you can take a screenshot of this so you can use it for your own sake. But yeah, again, A, B, D, antigen here, antibody here, antibody here. Okay. Let's see. I go and buy these. I put the antibodies to their specific antigen. Here, here, and here. And nothing happens in vial A. In vial B, a reaction occurs. And in vial D, agglutination occurs as well, which means that this guy is B positive. He can receive blood from B positive, obviously. Okay. Oh, sorry. No, he can receive blood from B positive. He can receive blood from O positive, because O gives to everybody, and positive to positive. He can receive from B negative, okay, since negative can give to positive and negative, and O minus. Okay, easy. Who can he donate to? He can donate only to B positive and AB positive. Easy. Okay, last two blood samples. We got A, B, D, A, B, D. Okay. I go and buy antibody, antibody, antibody. I put them in. Nothing happens for all three of them. I can conclude this guy is what? This guy is A? Not nothing occurs. A reaction occurs in all of them, actually. Okay? Reaction occurs in all of them. So this red blood cells start to clump and agglutinate in all of them, which means this guy must be AB positive. Let's change the color. AB positive, which means he can receive blood from both A, positive and negative, B, positive and negative, and AB, positive and negative, and O minus, and plus, okay? This guy is the universal recipient, okay, while this guy is the universal donor. So who can this guy donate to? He can donate only to AB positive. That's it. Last one, and then we're done. A, B, D antibodies. I'll put them in here and in here. No reaction occurs. The only place where agglutination of red blood cells occurs in this new sample is in the D vial, which means this guy must be O positive. O positive can receive from A minus and A positive, right? Because, remember, blood type. Oh, actually, sorry about that. I keep getting these mixed up. He can only receive from O positive and O negative, because positive can give to positive, O can give to O, and negative can give to everybody. Okay, so who can this guy donate to? Well, he can donate to pretty much everybody that is not negative. So he can give to O positive, A positive, B positive, AB positive. Okay, yeah. Okay, everybody. That's going to finish off this video about blood typing and how transfusion reactions actually work. Okay, I hope this video was useful, and I hope this kind of practice here made sense. And if you learn something, if you enjoy it, then please go ahead and like, comment, and subscribe, and I will see you in the next one.